Hello, this is Ed Chapman, and this is part 3 of our tour of the important parts of a eukaryote cell. We're going to talk about the cytoskeleton now, which, just as the name sounds, it's the structures that support and give a cell its shape from the inside. So it's the skeleton of microtubules, microfilaments, and intermediate filaments that hold up the inside of a cell. Now, microtubules are made of protein subunits of material called tubulin, and they fit together into a spiral pattern to build a hollow tube-shaped structure called a microtubule. And microfilaments are a lot smaller than microtubules, and intermediate filaments in size are somewhere between the microtubules and the microfilaments. Microfilaments are very tiny structures that give the cytoplasm its shape, and they're very difficult to see unless the cell has been dyed with a fluorescent dye that makes them show up, as you can see in this micrograph. Intermediate filaments are made from material like keratin, which gives the cell elasticity and also contributes to its shape. It also holds the nucleus in its location in the cell. The cytoskeleton also includes a mechanism for moving materials around inside of the cell itself. You may have seen in the plasmolysis video the chloroplast moving in a circle around the vacuole, or you may have seen in a popular web video, which I'll put the link on my website, where a vesicle is being pulled along a microtubule by these motor molecules, which actually walk along the microtubule, pulling the vesicle along with them. The cytoskeleton also includes centrioles and flagella and cilia. Centrosomes and centrioles are involved in the organization of the mitotic spindle, or the spindle apparatus, that's used to move chromosomes around during cell division. And cilia and flagella are motor devices that are used, or motor organelles that are used to allow some animal cells to swim or move through liquids. The next part, or the final part of our tour of the cell, is going to be what's going on at the surface of cells and at the places where cells touch each other, which we'll call cell junctions. And we're going to look at plant cells first. Plant cells, of course, are distinguished by their cell walls, something that's absent in animal cells. And the cell walls are made up of parts. There's the primary cell wall. Here you can see the primary cell wall of one cell to the right. We'll label that cell 1. And then the primary cell wall of cell 2 to the left here. And you can see how they come very close, but they don't touch. In between is an area called the middle lamella, which can be made of a material called pectin, which is very sticky, and it kind of glues the two primary cell walls together. Now, inside of each plant cell, you have secondary cell wall material, which can be very thin or very thick, depending on the function of the plant cell that you're looking at. For example, plant cells that are part of wood have very thick secondary cell walls. Plant cells also can communicate with each other through tubes that penetrate the primary and secondary cell walls and allow material to pass from one plant cell to the one adjacent to it. And these are called plasmodesmata. Plant cells also, because they're able to communicate with each other, when multicellular plants assemble cells in the tissues, we get emergent properties. We get something that can happen that wasn't possible before, because we've achieved a level of complexity that didn't exist before. So, for example, material can move from the roots of plants up to the shoots or the leaves of plants through a connected series of plant cells in a tissue called phloem, or inside of dead cells in a different tissue called xylem, which we'll talk about later when we talk about transport in plant cells. But this emergent property of transport is something that happens because of the ability of a multicellular plant to communicate cell to cell and move materials around. Animal cells, on the other hand, are usually surrounded by a material called the extracellular matrix. Now, when you see the word extra attached to any word, it means outside of. So the extracellular matrix is the material outside of an animal cell's membrane that allows it to hold itself up or attach itself, adheres to other animal cells or other cells, and also maybe to move or to regulate it in some way. Animal cells The extracellular matrix in, for example, your skin contains a lot of protein fibers made of collagen, which attach or interact with the outer surface of the cells in your skin to hold it together. You may have heard about people getting collagen injections because as your skin cells age, they lose collagen and start to sag in relation to each other. Young animal cells or young skin cells in animals has lots of collagen, and there's lots more connections in the extracellular matrix. So young skin tends to be bouncy and stretchy and doesn't sag or have as many wrinkles. Proteoglycans interact with, or what's actually attached to the cell membrane, and these interact with by literally tangling with the collagen fibers of the extracellular matrix to give some animal tissues more support. Okay, animal cells also have places where they connect more tightly to each other, or where they can actually exchange materials between them. And the first type of interaction here at the junction of animal cells is something called a tight junction. I like to think of these as staples because they hold the adjacent animal cells together very, very tightly, like a staple would hold a stack of papers together. And these tight junctions are what make some animal tissues very tough and difficult to tear. For example, your epidermal cells contain lots and lots of tight junctions, which make your skin flexible and movable but resistant to tears. Desmosomes are all locations where fibers are holding the two adjacent animal cells together. I kind of think of this as like they're being sewn together. And finally, we have pipelines connecting to adjacent animal cells called gap junctions. So animal cells interact with each other in three different ways: very tightly held together with tight junctions, or sewn together with material in something called a desmosome, or in places where they can actually exchange cytoplasm material through gap junction pipelines, so to speak. That ends our third part of the slideshow about cell parts, and I hope you've had a good tour of the cell.